Hi guys, this is Mrs. Polson. This is Unit 3, Segment 3, the last segment for this unit. It is called Cell Boundaries and Transport. You guys know the cell boundary is either the cell wall or the cell membrane. We're going to talk specifically about the cell membrane. This is the guy who lets things in and out of the cell. So we're going to talk about how things do go in and out. Here's a list of the vocab. Lipid bilayer. You might also hear the word phospholipid bilayer. That is another name for the plasma membrane or cell membrane. You got diffusion, equilibrium, concentration gradient, osmosis, isotonic, hypertonic, hypotonic, facilitated diffusion, active transport, endocytosis, exocytosis, phagocytosis, and pinocytosis. Those are all the vocab. Those are the power words you need to know. There's a benchmark of explaining the role of the cell membrane as a highly selective barrier, including the transporting things in and out through diffusion, osmosis, and active transport. All right, get ready to write. Here we go. Lipid bilayer, also called the phospholipid bilayer. This is another name for cell or plasma membrane. We've learned this already as an organelle. 2 layers of lipids. It's a flexible barrier for the cell and allows things to go in and out. Please draw this in your notes. These are the glycerol heads. If you guys remember the lipid, it's got a glycerol head and it's got fatty acid tails. This is just a small chunk of the cell membrane. This would go around and wrap around the entire cell. You will need to know the hydrophobic part of this cell membrane, and that is the tails. You will need to know that. It's on the practice quiz. It could be on the test as well. So this area right here is hydrophobic. It's not water-loving, so therefore it organizes itself away from the water that's outside this membrane or inside the membrane. So, or within the cell and outside the cell. So it will organize itself together. These tails, bilayer, there's 2, bi means 2, it's 2-layered lipids. And then the hydrophilic end, philic, it's water-loving, so they're organized toward the water. The heads, the glycerol heads, are organized toward the water. You need to know hydrophobic, afraid of water, oriented inside. Hydrophilic, likes water, organized outside. This is kind of an artist's rendition of what the cell membrane kind of looks like. So you have your cell, it's got the glycerol heads, and then it's got the fatty acid tails, hydrophobic tails, and then heads down here too. You've got water on both sides of the cell. There's water in the cytoplasm, and then there's water outside the cell too. Now, how do things get in and out? And that's the focus of this segment. Diffusion by itself is just molecules moving from a higher concentration to a lower concentration. So up on top of a hill, rolling down the hill, okay? No energy is required for diffusion to happen, guys. This is when you walk down the hall and somebody has spilled cologne. It happens all the time, and that cologne is concentrated at the locker that it broke, okay? But over time, it will permeate, diffuse, and move outward. That smell, those fragrances, will move outward. That's diffusing from where it's highly concentrated where it broke to where it's not so high concentrated, and it will just kind of move out until it reaches equilibrium. Now that's an example that's not talking about a cell, but we do have diffusion happening with cell membrane. So here is a beaker that's got a semi-permeable membrane, so similar to like a cell membrane. And the beaker has a whole bunch of things dissolved in this water on this side, not very much over here on this side. We'll give it enough time, and this is going to go ahead, these particles are going to go ahead and move across that semi-permeable membrane. Permeable means it can pass through. Semi means it's partial to certain things. So this red is allowed to pass through, and eventually it'll reach an equilibrium, which is what you see here. I would draw this and label it diffusion in your notes. And I would label this one equilibrium. So equilibrium is when the concentration is equal in all parts of the solution. So there's really no net movement. Molecules are always going to move back and forth, but there's no overall net movement anymore. You're not going to have mass amounts going over here or mass amounts going over here. You're going to have this guy might move and this guy might move. You're going to have movement back and forth, but you're not going to create any net movement, any overall numbers movement. Okay, so equilibrium is the example that's shown here, not here. Which one is at equilibrium? So we have this one, we have the middle one, or we have this one. Here we're looking at a bilayer, lipid bilayer, cell membrane, and which one, 1, 2, or 3, is showing equilibrium? The answer is this side. Yes, this blue is moving across, and this one is too, and they're moving back and forth, but there's no net movement. There's no overall because they're equal concentrations on both sides. Concentration gradient, you guys, this is going to be when you have a balance of concentration in this solution. Actually, it's when you don't have it. It creates a gradient. So when you have more on one side versus the other side. So things will always move from higher to lower. And if they don't, if they move from lower to higher, that's going to require energy. And the first several ways of transport that we're going to talk about do not require energy. So the concentration gradient itself is enough push that there will be movement of particles. So we have a situation where we have a solute, which is red in color. It's concentrated over here on the side of the beaker. Here is water. This is solvent. The solute over time begins to move, and the blues are always moving as well. And over time, this is what it looks like, and this is called, starts with an E, equilibrium. So there was a concentration gradient of this red over here. There wasn't so much with the blue, but with red, it's really concentrated over here. So therefore, there's going to be a push of movement just naturally through diffusion to move through and equalize itself, reaching equilibrium. Osmosis. This is the special diffusion of water. So if we're talking about diffusion of anything else, it's just diffusion. But if we're talking about diffusion or movement of water, we have a brand new name, and it's called osmosis. You should say it out loud wherever you're at, osmosis, because you're cool like that. So, but because this is diffusion, there's no required energy. It's using the concentration gradient to drive the motion. Water will always move from higher to lower concentration through osmosis, always down that hill. So here's a situation. We have two beakers or containers, and the pink is a solute. Some kind, it could be salt, could be sugar, could be whatever. This semi-permeable membrane is not permeable to the dots. So the dots will not move. That solute would not move. So only water can move. Well, if you look at the fact that there's more solute over here and less solute over here, we can switch our thinking and say, well, if there's less solute here, that means there's more water on this side. And there's more solute here, so that means there's less water concentration on this side. Okay? So water will move from higher concentration to lower concentration. Since there's a higher water concentration over here, it's going to move over into this chamber. And over time, this is what we will see. Water indeed moved over into this chamber. It actually went where it was more concentrated to where it was less concentrated. And you guys, this is where you can get caught up and confused. With osmosis, we are looking at the concentration of water. So if you are told the concentration of solute, you have to flip that and think, all right, if solute is 20% of the solution, that means water must be 80% of the solution. You're going to see it both ways, and that's going to be tricky. We're going to talk about that in class. So through osmosis, we have different types of solutions. There's isotonic, hypotonic, hypertonic. Isotonic, iso means equal. So this is a solution that has equal concentrations of water outside and inside. So in this situation, isotonic is the one in the middle. we can see that the solute, which is green, is equal in number, and then the solute over here is equal in number to what's inside the cell, and so there's no, that means the water concentration is also the same. So there's no net movement, okay? Now, with this situation, I'm going to explain these pictures right now, but the next couple of slides are going to have you define it. But with hypertonic solution, water will move outside the cell. Now let's look at this and try to make sense of why we would have water movement going outward. So what we see, the green dots, is the solute, not the water. So the solute concentration is low on the inside, which means the water concentration is high on the inside. On the outside of the cell membrane, the solute concentration is high, but the water concentration is low. So we care about the water concentration when we're talking about osmosis. Where is the water going to go? From high to low. So from inside the cell, it's going to move outside the cell. The cell will actually shrink in a hypertonic solution. Let's look over here. Hypotonic solution. Hypo means that water is going to move in. Let's talk about why that makes sense. Here we have one little particle, one little solute. That means there's a lot more water because there's only one solute. That means there's a lot more water concentrated out here. Because there's 6 solutes in here, that means there's less water concentrated on the inside. Water moves from high to low. So it's highest out here, lowest in there. It will go through the membrane and swell up this cell. If you didn't understand that, let's watch carefully on these next slides. Hypertonic, H-Y-P-E-R, tonic solution. This is going to be when there's a higher solute concentration outside the cell, which means there's a lower water concentration outside the cell. That means there's a higher water concentration inside the cell, and so the water is going to move out. So hyper is over here, the water is going to move out. Another way you can think of it is water wants to dilute. Water wants to dilute this solution. So where the solute is high, water wants to move to dilute it. So it's going to move toward where there's more solute, which means there's less water. Okay? This is something you really do have to play with and practice with. Hypotonic. Here's a little trick. Hypo. See this O? Picture that as the swell. As the cell. So the O is going to take in water and swell up to make the O. So you have the O shape. It's big. It's bulging because water is moving into the cell. So now let's talk about the definition. This is when we have diluted or lower solute concentrations on the outside of the cell, which means there's a higher water concentration outside the cell. Water is going to move in because it's higher outside. It's going to go to where it's lower. So water will move in. Looking at this picture, and I would draw it in your notes, you have not very much solute outside. That means there's higher water concentration on the outside. Water is going to move in to try to dilute this right here. There's a lower water concentration on the inside. So it's going to go from where it's higher to where it's lower. It's going to move in, swell it up, make it O, hypotonic. Here's some examples. For hypotonic situation, you have water moving into the cell. It's going to lyse or split or break. Here is this is an animal cell. This is a plant cell, so it kind of bulges the cell wall out a little bit. Isotonic, there's no net movement in or out. There's always movement in or out, but there's no net movement one way. So in both situations, so the cell stays the normal shape. Hypertonic solution, this is when you have, like, you put salt on a snail. Okay, if you put salt on a bloodsucker or something like that, the water will come out. Okay, I had to cut this off. I had to cut the recording short a little bit because in 20 seconds this recording will stop. I can't go over 15 minutes. So this is going to continue on a second video. Watch for the second video for segment 3, unit 3.